Global Food Regulatory Science Society. GFORCE mission includes knowledge generation and dissemination, underpinning food regulatory decisions, in support of food control systems. Consistent reliance on food regulatory sciences enables robust food control programs.
Good morning, good evening, good afternoon, everyone, and welcome to today's webinar on methods of analysis supporting halal food certifications. My name is Ruba Goussou. I'm currently working as a food safety expert at the Jordan Food and Drug Administration, JFDA, and a member of the Global Food Regulatory Science Society, GFORCE. I'm pleased to be with you today and to moderate today's fifth webinar organized by GFORCE as part of its Food Regulatory Science webinar series, and to have participants from all around the world today. Before I introduce our speakers and the topic, please note that Arabic interpretation is available for this webinar as well as live broadcasting through GFORCE YouTube channel. Additionally, we encourage you to submit your questions throughout the presentation via the chat box. We will have a dedicated Q&A segment following our speaker's talk to address your questions. And here I would like to invite Dr. Eric Connings, co-chair of the Committee on Sections of AOAC International, to share with us his introductory remarks. The floor is yours, Dr. Eric.
Thank you, Ruba, and greetings to everyone online. My name is Eric Konings. I'm past president of AOAC International and currently co-chair of the Committee on Sections. For those who don't know, AOAC is an independent, non-profit membership association of analytical science professionals in government, industry, and academia all over the world, and in standard developing organizations. On behalf of AOAC International, I would like to welcome you to this webinar. I would like to acknowledge the organizers. I think it is the first time that two of the in total 15 AOAC International sections joined forces to organize this webinar for their common interested audience. Secondly, I would like to emphasize on the importance on communication between the regulatory and analytical sciences community, because you need both in regulatory decision-making. I hope more of these joint webinars will follow in the future. I wish you an informative and interesting webinar ahead of you. Thank you very much.
Thank you. Thank you very much, Dr. Eric, and appreciate all your words and encouragement. And I would like to invite now Dr. Xingping Hu, President of Southeast Asia section of AOAC International, to share her thoughts with us. The floor is yours.
Thank you, Roba.
Welcome.
Good morning, good afternoon, and good evening, everyone. Welcome to the webinar. First of all, I would like to thank Dr. Amy Koso for inviting AOAC Southeast Asia section to co-support this GFORCE webinar together with Arab section. This webinar is very relevant to the analytical communities in Southeast Asia where halal food certification is widely applied. The section members here share the common interests with the analytical communities in the globe, regarding halal food certificate regulation in different countries and method of analysis supporting halal food certification. It facilitates the compliance for food safety and quality in international trade as well. It is our honor to co-support this great webinar. We are very glad to have Mr. David YULIANTO from SIG Laboratories in Indonesia, one of the AOAC Southeast Asia Section's organization members, to present on the halal food certification in Indonesia and advanced analytical techniques in his labs. So I believe that Mr. YULIANTO's presentation from Southeast Asia's perspective will be beneficial to the audience. I wish the webinar a success and hope everyone enjoyed the talks from the distinguished speakers. Thank you.
Thank you very much, Dr. Jinping, and appreciate all your words. I would like also to invite Dr. Amin Koussouf, President of the Arab section, to share also his introductory remarks. Dr. Amin, please.
Thank you, Dr. Ruba. Good morning, good afternoon, good evening, everyone. So also, it is my pleasure to welcome each of you to this important webinar on methods of analysis supporting halal food certification. The topic of halal food certification holds a particular significance for the Arab region, where there is a strong and growing demand for assurance that food products align with halal standards. It is a matter that touches upon not only food safety, but also cultural, religious, and ethical values that are integral to our communities. Today's webinar brings together stakeholders from various regions, highlighting the importance of interregional collaboration in advancing and harmonizing halal certification standards. Together, we will explore analytical techniques and methods that are crucial in upholding halal standards, ensuring that our food supply chains meet the high standards expected by consumers across the Arab region and beyond in the world. Thank you for joining us for this crucial discussion. I am looking forward to these insights that will emerge from today's presentations and to strengthening our collaborative effort in the pursuit of excellence in halal food certification. I would like to thank Southeast Asia section for the support. I would like to thank AOAC International for the continued support. I would like to thank our esteemed speakers, and thank you, Dr. Ruba.
Thank you.
It's a collective effort and contribution from all volunteers in the past few years. We appreciate everybody's effort in the past few years for every volunteers that you contribute in a different way.
We eagerly anticipate the knowledge and insights that will be shared, which will guide us in understanding the procedures that can lead to more reliable outcomes and consequently bolster consumer trust in our food supply.
It is a great honor to be here today at the third annual conference of the Association of Official Agricultural Chemists Southeast Asia section.
We have increased our collaborations with the industry partners to strengthen our efforts to ensure food safety and security for all Filipinos. We align all our programs, projects, and activities to contribute to attaining the 2030 Sustainable Development Goals.
What is the sample you're working on? What are the analytes? And more importantly, what is the detection limits? With these three questions, we will be able to kind of offer you, a preliminary solutions on what you can do.
Looking at this audience here, I can say we are still quite vibrant and still relevant organization to the international food safety professionals.
So we are trying to modify our standard extraction method specifically for our heat-treated sample, and we are able to get a very good, closer to a good recovery, closer to or an improved version of our results.
How individual labs can validate their testing of larger sample sizes. and that there is a transition period through 2027 for the generation of the necessary data by the individual labs concerning testing of larger sample sizes.
So the government creates policies and food standards in ensuring food safety, and laboratory testing is an integral part of this system.
By using a CRM, from time to time, it allows the laboratory to monitor their own internal drift, iterate that scientific evidence are needed before establishment of policies and regulations.
Legislation requires the absence of certain regulatory components, then the LOD must be studied more rigorously.
Many capabilities that we can do, specialty in food, feed, and also another consumer good samples.
Actually brought to the attention of many foods companies that they want to mitigate this not only in the final food product, but it actually involves the whole food production supply chain.
So the Metrology and Chemistry section conducts method development and validation of chemical methods, especially the emerging contaminants. Then we produce reference material, provide a proficiency testing scheme, and offer other services such as training for chemical testing laboratories.
With this two-day conference, I really hope that you make some friends because I'm doing that myself. Thank you for your sponsorship and thank you for your participation.
We are all consumers, so definitely we are exposed to the potential risks. And the main concern is always the endocrine disruptors ability, because they can mimic the hormones in our body, so that can pose public health risks.
Populations serving, like for example babies and infants, you have to ensure that what is actually in the product and labeled is also in there. They have to meet regulatory requirements and also ensure nutritional claims.
Which are the food sample matrix that is of your top concern as well? And to our surprise, herbs and spices actually came out on top and consists of more than 80% of the responses when it comes to analytical challenges.
Now, the DOST ITDI conducted a study to develop and validate a method for the accurate determination of methyl and propylparaben in tomato sauce using HPLC PDA. This study is actually part of the ongoing project of the Institute on the standardization and validation of chemical test methods on food safety parameters among the DOST laboratories.
Having a fast-growing economy, we face a lot of issues, and one of them is about the agricultural intensification. This intensification leads to the overuse of pesticide and drugs.
Over the past few decades, we have been supplying total solution for analytical reagents, which is high quality, dedicated to support and a wide range of lineup.
Thank you very much for your support, and most of you have supported our annual meetings in the past few years, and we are also glad to see a few new supporters this year, and we will continue to try our best to make the future conference successful.
Halal food certification is essential in ensuring that food products meet specific requirements in composition and preparation methods in line with the beliefs and ethical standards adopted by Muslim consumers. This certification Process involves verification, various stages of production, relying on rigorous analytical methods to ascertain purity and authenticity of ingredients. In this webinar, experts will provide an overview of the key characteristics of the regulatory framework for halal certification, followed by detailed presentations on advanced analytical techniques used to verify food composition. The techniques highlighted include real-time PCR and LC-MS for detection of porcine, DNA, and gelatin. Headspace, GCFID for measuring methanol and ethanol in beverages. Additionally, the session will address global challenges in harmonizing analytical standards and regulatory requirements for halal certification, offering insights for professionals in food safety, quality assurance, and regulatory fields. Today, we're fortunate to have two distinguished speakers who will share their insights on methods of analysis supporting halal food certifications. Thank you all once again for joining us today. It's truly a privilege to be moderating this webinar alongside such esteemed experts. Their expertise and perspectives on this critical topic will help us better understand the latest advancements in halal food certification. Our first speaker is Dr. Silvia Dominguez. Dr. Silvia is a food engineer specialized in food safety, risk assessment, allergen management, and data modeling. She holds a master and PhD in food science from Rutgers University, USA. She currently works as a research associate at the Food Risk Analysis and Regulatory Excellence Platform, hosted by the Institute of Nutrition and Functional Food of University Laval, Quebec, Canada, where she manages academic research projects as well as food safety capacity building initiatives for competent authorities at the international level. And with that, I'll hand it over to Dr. Silvia. Dr. Silvia, please, the floor is yours.
Hello, everyone, and thank you, Ruba, for the introduction. So I will start the webinar today by providing a general overview of the topic we discussed today, which is methods of analysis supporting halal food certification, before our colleague, Mr. YULIANTO from SIG, presents the more technical part of the webinar. Next slide, please. So first of all, let's discuss what is halal food and why certification is important. Halal food actually represents quite a large portion of the global food market, almost as high as 20%, which is not that surprising if we consider that halal food is consumed not only by the very large Muslim population around the world, but also by consumers that are looking to purchase ethically produced foods. And this is a trend that has gained popularity in recent years. The term halal means allowed or permitted and wholesome in Arabic, and is part of the Islamic principle that emphasizes purity, cleanliness, and health. Although halal food specifically does not have a universal definition, it's understood that this food must be produced according to Islamic law. In fact, halal food is not defined by what it contains, but instead by what it must not contain. So ingredients that are not allowed under the Muslim faith, also called haram ingredients, must not be present. And these are, for example, pork and its derivatives, blood, alcohol, and, for example, meat of animals that have not been slaughtered according to Islamic law. So these forbidden ingredients must not be intentionally added to the food, and also the food must not have been in contact with them either at any point on the supply chain. So these are generally recognized basic criteria for what is halal food. However, different Islamic groups may interpret specific points differently or may have additional criteria, which is actually a challenge for the development of a harmonized halal food standard, as we'll see in following slides. Next slide, please. So yes, as I was saying, there is currently no unified halal food standard, meaning there is no harmonized official definition that would be valid in all jurisdictions. However, there are several countries and regions that do have their own standards related to halal food. For example, as you see in the table here, countries like Singapore, Malaysia, Indonesia, Thailand, Pakistan, the Netherlands, etc., have this type of standard. And we also have regional standards, like for the Gulf countries through GSO, and also standards from Islamic organizations like the Standards and Metrology Institute for Islamic countries. And apart from standards, there's also guidelines. Guidelines are recommendations or best practices for the use of the term halal or for the production of halal food. And these have been proposed by international organizations, notably by the Codex Alimentarius Commission and also the Association of Southeast Asian Nations. And if we see Brunei also, they have standards and guidelines for halal food. So all these documents, standards, guidelines, etc., cover different aspects related to halal food, some of which actually go beyond only definitions of the term. For example, apart from general requirements, some of the topics that are covered in these documents include quality and supply chain management systems, requirements for certification bodies, requirements for testing laboratories, etc. And there are also norms specific to certain operations like slaughterhouses, meat processing facilities, hotels even. And there are documents specific to certain products of interest like gelatin. Next slide, please. So apart from variability on what different countries' standards will cover, the interpretation of certain common topics like slaughter, stunning, seafood, insects, etc., may also differ. So I want to showcase some of these differences with a few examples. The first one would be about slaughter. Slaughter, for instance, is one of the most important items related to halal food. So in general, under Islamic law, animals must be treated humanely at all stages: housing, transport, even slaughter. For example, there are indications or requirements for young animals to grow up closely to their mothers. There are also requirements for the trucks transporting the animals not to be overloaded, etc. And the Slaughter Act itself, generally it should be conducted by a Muslim person using a knife in the neck region, and the animal must bleed out. So these are general criteria for what is slaughter under Islamic law. But there may be different interpretations or specifications depending on the Muslim groups. For example, this table highlights that regarding who can perform the Slaughter Act, in the jurisdictions listed here, it is specified that it has to be a Muslim individual, except in GSO countries where if a Muslim is not available, a person of another faith may conduct a ritual. And also we see requirements about the age of the slaughterer in Indonesia and Malaysia, but not in other jurisdictions. So this is just one tiny example of how we can see things vary among the different standards in the different countries. There are several other differences related to slaughter in particular. For example, there's a lot of variability regarding the use of stunning methods. But the point I wanted to make here is that these differences are due to variations in interpretation by different groups. And this is actually something that is acknowledged even in the Codex text related to the use of the term halal. Next slide, please. Another interesting example on different interpretations is related to insects. We can see in this table that locusts, a type of grasshopper, are allowed in all jurisdictions listed, but there are differences with respect to ugly and not ugly insects, which, as you can imagine, is kind of subjective. And then more relevant to our topic today on analytical methods, we have another table here showcasing differences related to permissible alcohol levels. So in theory, alcohol is not allowed, as we saw before, it's a haram ingredient. But we see different countries differentiate naturally formed versus industrial ethanol, with some allowing up to 0.5% of industrial ethanol and up to 2% of natural ethanol. Next slide, please. So bottom line, what we saw so far is that we have only a broad definition of what is halal food, and we have multiple interpretations, so therefore we have multiple standards. Let's discuss now certifications. Who can certify if a given product can be labeled as halal? Well, since we don't have a unified halal standard, we don't have a unified halal certification either. Instead, we can find many halal certification bodies. For example, only in Malaysia, there are more than 80 halal certification bodies that come from more than 40 countries. And that's a lot if you compare with more traditional food safety certifications. There's also no harmonization on how these certification bodies operate, meaning which criteria they apply to certify that a food is indeed halal. Some follow country-specific standards, which we just saw can vary a lot, and some others have their own criteria. Next slide, please. With respect to verification, there are also a number of challenges. As you can expect, proving that a food meets all the religious criteria to be considered halal can be very complex. So an important verification tool that we can use are audits, which should be conducted on site to ensure the ritual aspect is respected in the case of slaughter, for example, but also to verify the operation has measures in place to prevent cross-contamination with haram ingredients. And this should go beyond the actual production site to cover as much of the supply chain as possible. Certifications could also be a tool to verify that the food is indeed halal. However, they should be verified with audits, especially considering the great variability expected between certification bodies that we mentioned before. And usually, audits or certifications are combined with laboratory analysis, which are extremely important, specifically when investigating the conformity of suspected samples. Notably, analytical detection of pork and alcohol are widely used as part of halal food verification requirements. In the table you see here, I listed some very basic examples of foods that may be subject to analytical testing for halal verification. I'm mentioning, first of all, halal meat products. For example, let's think of sausages. They may require testing to confirm absence of pork meat, which may be present in this type of product due to cross-contamination, but maybe even fraud because pork is cheaper than beef. And we're not only talking about pork meat, we also know pork derivatives are forbidden. For example, pork gelatin, which is an ingredient widely used in the non-halal confectionery industry, may be found in this type of products like candy or marshmallows. And then we also have, of course, alcohol, which would in some instances not only need to be detected, but also quantified to establish if the food can be considered halal or not. And this could be the case in products like beverages, of course, and fermented sauces. Next slide, please. So our next speaker, Mr. YULIANTO, will go much deeper into the analytical methods part of the webinar. But I wanted to showcase some differences between what we find in the scientific literature versus commercial methods. In the scientific literature, we can find a variety of methods that have been used for haram ingredients. For example, for animal species identification, so we're talking about pork and its derivatives, we're mainly talking about DNA-based methods like PCR. But then we can go into more specific applications. So for example, to establish if fats are of animal or vegetable origin, we see in the table methods like infrared spectroscopy, liquid or gas chromatography, coupled or not with mass spectrometry, or even electronic nose. Then we have applications for protein composition. So for example, to see if protein comes from pork or from other animals. We have, of course, SDSPH, ELISA, but also infrared spectroscopy and chromatographic techniques. And even more specific applications, like, for example, to establish if a sample is of natural or synthetic origin. We find studies that have used advanced techniques like magnetic resonance or isotopic analysis. And finally, for alcohol, scientific studies have used gas chromatography, electronic nose, and proton transfer reaction mass spectrometry. So it can get very technical and complex, at least as far as scientific applications go. In the next slide, I wanted to mention that among commercial methods, on the other hand, we can actually find rapid alternatives. For pork, for example, there's of course ELISA for the detection of proteins or PCR for DNA, which are the most common. And they're usually quite good to detect their target analytes, but quantification is not always as efficient. They both have their own challenges, especially when dealing with highly processed food matrices where protein or DNA denaturation may have occurred. And also both methods have advantages and disadvantages with respect to time and cost. But I will leave that to our next speaker. And for alcohol, rapid methods based on enzymatic reactions coupled with color indicators are available for ethanol, propanol, methanol, etc. But more advanced techniques like gas chromatography are also used. Next slide, please. So that was a brief introduction to the topic. Now I would like to give the floor to Mr. YULIANTO from SIG to dive deeper into the technical part of the webinar. Thank you.
Dr. Silvia, thank you very much, Dr. Silvia. And now Dr. Dawi LAKSONO will be our second speaker. He is the General Manager of SIG Laboratories, which are laboratories that are state-of-the-art technologies and the most advanced scientific technologies for an excellent scientific-based solutions, accurate, precise, and consistent in all products testing and developments. Doctor, the floor is yours. And
okay, thank you, Ruba. Is my voice clear?
It's very clear.
Okay, thank you. I will share my slides.
Very good.
Okay, thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, good afternoon, and good evening in Indonesia. Thank you very much for this occasion to SIG Laboratory to speak in webinar called Ensuring Halal Products by Laboratory Testing. I would say thank you for AOAC Southeast Asia, Dr. Eric, Dr. Sinping, and also AOAC Arab Section, Dr. Amin, and also my colleague, Dr. Silvia, and also for moderator, Dr. Ruba. Thank you very much. I will speak about this maybe a little bit, just share our experience regarding the halal testing in Indonesia. First of all, I will maybe introduce about our laboratory. We call it PT Saraswanti Indogenetek or SIG. SIG is one third-party laboratory in Indonesia. We established in 2001. And now we have not less than 450 employees. And also approximately for average, we receive more than 2,000 samples per month and 150 tests per month. And also, beside Indonesia, we already experienced to work together with our customer in across the globe, from USA, from Europe, UK, and also from Asia and also Southeast Asia. And also, we already accredited by CAN or accreditation testing body in Indonesia for ISO 17025 or good laboratory practice, and several accreditation for GMP Plus for feed that our customer exported to Europe, and also for the designation for ministry in Indonesia, and also we also recognized by Ministry of Health from Japan and also European Union for testing in mycotoxin for nutmeg. And also proudly we are member also in AOAC International in Southeast Asia, and also we join in Halal Consortium in Indonesia held by BPJPH. That one is Halal Product Assurance Organizing Body in Indonesia, and several member in cosmetic and also laboratory for cosmetic testing and also food testing and also for food and beverage society in Indonesia. And just our testing service, but mostly 80 until 85% our samples is food and feed, and we also dealing with cosmetic, pharmaceutical, and etc. You can see all our capabilities in our website www.siglaboratory.com. You also can see about our accreditation scope and all our capabilities and all social media in Instagram and also in LinkedIn. And this occasion, I will speak our outline is about an introduction, halal in food industry around the globe, and food halal assurance, and halal in Indonesia in our perspective as a third-party laboratory, and also the laboratory positioning in halal certification, our testing service for halal, and also challenge and recommendation that we need to discuss with you. We need also your point of view from maybe in Arab section and also United States and Canada and so on. First of all, about the halal in food industry around the globe, and halal maybe my colleague Dr. Silvia has already explained about the halal is the permissible in Islamic law in Sharia, and the concept applies to various aspects of life guidelines for permissibility in dietary and lifestyle for Muslim. And also, in addition to being halal, the food consumed must also be toyib or good, and which is beneficially for the bodies and does not conflict with the command of Allah Subhanahu wa Ta'ala. And from the Quran in Surah Al-Baqarah, verse 168, and also from Surah Al-Maidah in verse 87. And non-halal, it may be for the beverage that contain alcohol or ethyl alcohol, blood, carnivorous animals, and also dead meat. It's meat not slaughtered according to Islamic slaughtering rites, immolated food, and also swine and derivatives. And sometimes that alcohol is contained in several fruits that maybe in Indonesia is very famous in durians, in Southeast Asia, yeah, Malaysia, Thailand, Philippines, and also Singapore. Durians called king egg fruits, and in the fact, durian is contain alcohol, but uh, in the in the law in Islam in Surah Al-Araf verse 157 is permit for them what is lawful and forbids to them what's impure. So durians from the Indonesian Ulama Council, durian is still halal, but it's contain alcohol. It's from the plants, and also sometimes in several countries there's a fermented food. We call it in Indonesia is tapai, is made from cassava and also from rice, sticky rice. Yeah, it's also fermented and also uh, containing alcohol, but it's still halal because it's not a khamar. Khamar, its mean is intoxicating drink made from fermented grape juice, so it's halal to consume. And also about the surah from Quran from Al-Araf verse 31 that eat and drink but not excessive. And also from hadith from An-Nawawi, there should be neither harming nor replicating harm dirar. So it's mean the Prophet Muhammad SAW also told that we must eat when we're hungry, we stop when we full, and so on. So halal is many perspective in Islamic way. And from our perspective, that's halal food in global market that's mentioned is increasing, and also from this Anish and Kader in 2022, maybe the Muslim population will around 3 billion in 2060. And also from the Asia Pacific food industries, that the projected growth rate of halal is 9.1%, reaching USD 4.1 trillion by 2028. So halal in global market is so promising around the world. And from the next outline for food halal assurance, that for urgencies for halal product certification, that the one is the customer trust, especially for Muslim countries or Muslim societies, and also market access for around the world, especially the food or consumer good producer to export the products to Muslim countries. It needs to urgency for halal certification and also for advantage, competitive, and also quality assurance to make sure about the safety for Muslim consumer, particularly when it comes to consumption of market products. And this is the global halal certification bodies around the world, in from Australia, Sri Lanka, Malaysia, and Jakim, Singapore, United States of America, and also Turkey. And also, as we know that we have the standards or methodology institute for Islamic countries, that standard that also our government in BSN or Badan Standarisasi Nasional or National Standardization Agency of Indonesia already member of SMIC. So maybe in the future, our agency or BSN will conduct some regulatory or mandatory. We have SNI or National Standard Indonesia regarding for halal accreditation in specialty in Indonesia. And for our perspective, halal in Indonesia, and as we know that Indonesia is the country is the largest Muslim majority population, more than 80 until 85% our citizen is Muslim. So it's great potential in the halal industry, especially in the halal food, halal tourism, halal fashion, halal pharmaceutical, and halal cosmetic. And Indonesian government is also aim to make Indonesia the world halal center and the pioneer of halal industry globalization. And this is the regulatory framework in Indonesia about the law, about the government regulation in 2019, 2021. And now maybe about the halal certification body, we have majorly in 3 steps. We have BPJPH, or we call it Halal Product Assurance Organizing Body. We have LPH, Lembaga Penjamin Halal, or Halal Inspection Agency. That's usually connected with SIG or our lab to conduct it about inspection or also testing for halal certification, and also about the Indonesian Ulama Council to determining a product halalness through fatwa in religion or Islamic way. So these 3 steps is the major things that will be conducted if we want to certify halal in Indonesia. 3 steps from BPJPH, from LPH, and also MUI. And we have 2 steps actually for halal certification process flow. This is for the normal with applicant. Now we can do it in online, proposing the product for halal certification in BPJPH website. And the BPJPH will checking completeness of our document and determine the LPH or halal inspection agency. And the halal inspection agency will do or conduct the inspection, or also the auditor will see if the product or whole the process will conduct just only for the inspection or need to laboratory testing for make sure all the products is halal. And after that, the Indonesian Ulama Council will determine halalness to product through and halal fatwa. And then for halal certificate is issued by Halal Product Assurance Organizing Body or BPJPH. And the second step is self-declare. Self-declare is usually conducted by small or micro business in Indonesia. Usually we have several things to do with create isi halal according to proposing their product of halal certification. We also have process product halal assistance, or a person that assists micro and small business actors to fulfilling the requirement for a product halal statement. Usually they came to our residence community in Southwest community in our neighborhood or our residents to conduct the webinar, conduct the seminar, conduct about the things regarding the halal statement for self-declare, and the data verification and validation from applicant will be approved in BPJPH, and then we not go through from the halal inspection agency, but directly to MUI and also BPJPH for issuing the halal certificate. And usually this is the same comparison because the regular and self-declare, because right now our government is have mandatory for halal certification, usually for large or medium or large scale industries. They have subject for fees, but in the micro business is right now is still free of charge, and also is engaging for applicant assistant for verifying and validation halal audit. So the micro business is now they very push for regarding the self-declare halal certification to make sure about the goal of the governments in 2026. All the micro and small business in Indonesia already certified halal from BPJPH. So right now, from the micro business, in 2024 in October is already mandatory, and the goal is 2026 all the micro and the small business will also have halal certification obligation for customer protection and business confidence. Yeah, that's the goal from Indonesia government, from Ministry of Religion in this proxy is from BPJP. And you see also we have the maybe in mobile phones or in the Play Store about the program for halal checker. The customer and also the micro business can conduct the halal certification from their cell phone to make the self declaration is more easier. And after that, we will talk about the SIG halal testing services. That why is the background that we conduct the services regarding the halal? Yeah, actually, all about the our customers that until 85% from the food and beverage industries. Usually, they have additive like from gelatin, and they have also coating. They also flavoring and encapsulation sometimes in their products. So it need to assure that all their raw material is from the halal raw materials. And this is so many food and in the industries that maybe can able containing non-halal substance, maybe in gelatins and also from lard and also from alcohol and also from this is soft capsule for pharmaceutical industry. That's we have so many products that maybe contains non-halal products in our market specialty in Indonesia. And we have three things or three kind of a method of analysis. First, we have qPCR. That's a technique for DNA identification for authenticate portion DNA. And we also have LCMS-MS, the technique for detection of specific markers as an indicator of the presence of portion gelatin. And also the third one, we have GCFID headspace, a technique for calculation alcohol content quantitatively, usually in ethanol, in sauce and etc. Matrices. And from the real-time PCR, this is the scope method that we also can get in our website in www.slglaboratory.com. This is the matrices that we already do the validation and also already in our accreditation scope in the meat sausage, in the instant noodle, sauce, meat, gelatin, cosmetic, and also pharmaceutical products like chondroitin. And also this is the example for metric recap in 2024. The 24% is pharmaceutical products. We have also the noodles. We also the packaging, cosmetics, raw material, beverage, meat, sauce, and etc. And why using qPCR? Maybe qPCR in some matrices they have high sensitivity. They're capable of detecting very low amount DNA, so it's very effective for analyzing sample with low concentration. And also, qPCR is highly quantitative and also accurate. The application process it can be monitoring, and the specificity of the analysis for the primer for portion DNA can differentiate between portion DNA and other types, reducing the risk of false positive result. And this is the procedure for the qPCR. First, the DNA extraction, and also the second one is denaturation, and then the annealing, elongation, and also detection regarding from the qPCR that we are conduct in our lab. This is the example of the raw data or qPCR analysis. This is for the positive control and the inhibitor control. And the red one is the sample of the positive samples, and the blue one is negative samples, and also the black one is negative control, and also extraction blank control, to make sure our testing is not have data that can be false positive or false negative. And the second one is the LC-MS or liquid chromatography mass spectrophotometry. And the principle of the method is detection of % of bovine specific peptide biomarkers using LC-MS qualitatively by comparing sample reading to controls. And why we use also the LC-MS? Because sometimes in specialty in matrix and gelatin, yeah, we have accreditation scope in matrix like gelatin, soft candy, soft capsule that already have a small amount of contained DNA. So if we use qPCR or PCR to detect the sample matrix from gelatin, is usually can be a false positive. So we already have also for LC-MS for matrix from gelatin base. And also the matrix recaps from 2024, we have samples from soft capsule, we have soft candy, we have a gelatin powder, we have marshmallow, and etc. And also gelatin is a mixture of polypeptides and also from extracted from skin, bones, and connective tissue of animals. And they have a wide range properties and functionalities, and a lot of things that can be used in raw material, especially in food, for stabilizer, for emulsifier, for thickening agent, and etc. And very wide use by food producer, specialty in also beverage in several products in Indonesia also. And also why is should gelatin test to for halal? Yeah, because gelatin also can be made from swine and also from bovines, but to make sure that the gelatin is from the halal substance, so we should to test for halal or maybe portion detection to make sure about customer trust, the customer belief, and also compliance with market regulation. And also we using LC-MS for the specificity, for the highly sensitivity, for the versatility, because they're regarding about the metrics that it's sometimes the compounds on the matrix is the DNA is very low. Yeah, it's very low, and the process is very long. So the DNA is cannot detected by qPCR. So why you using LC-MS with several matrices to make sure we not have a false positive or false negative result. And the procedure is first of all the extraction. We use extraction for peptide with enzyme, and we do the separation with material column with liquid chromatography system. We do the fragmentation detection with MSMS, and we do the identification for the presence of percent specific markers that identify based on their unique mass signatures. These are the samples of chromatogram that we produce in our lab. This is the samples chromatogram that have a positive result in porcine, and this is the samples that have the not detected or negative result in the samples in chromatogram. We have identification with two markers that made by QTOF and made by several testing with our software in our LC-MS. And the third one is the GCFID or gas chromatography flame ionization detector. And the principle is alcohol content, usually methanol and ethanol, will be quantified with GCFID using a standard calibration curve. And also headspace method are used for detection methanol and ethanol as residual solvent. And for the accreditation scope, we have uh, the scope for the beverages, alcoholic and non-alcoholic beverages. We have also for pharmaceutical product, for syrups, and also we have scope for chili sauce or condiment. Usually they check also for the residual solvent of methanol and ethanol. And also in the 2024 matrix recap, we have 63.4% in pharmaceutical products, usually for syrups. And also we have 13% for beverage, for cosmetic, sauce, raw materials, and etc. and other in 2.1%. This example about the chromatogram that we are produced in our GCs. This is the ethanol detected result, and also this is non-detected. This is internal standard and propanol to make sure our test is quite robust in our GCs. And also, why the beverage must be tested for halal? Because testing for ethanol content is used for the critical parameters used the beverage ingredients, because it can categorize as homer and intoxicant drinks. And when we took from the Quran, is from the Surat Al-Maidah verse 90, that it's said that Muslims is forbidden to drink homer. And also, all alcohol is non-halal, and based on MUI or Indonesian Ulama Council, the alcohol have two categories. First is the alcohol or ethanol produced by the Khamar industry. It's Khamar industry for alcoholic beverages. It also haram and also najis. And also for second one is alcohol for produce for non-Khamar industry. Yeah, usually we use for solvents in our laboratory, also used as a solvent and sometimes reagents, and it is permissible if it is not medically dangerous. And the procedure is we have three steps. One is the dilution, dissolving sample using a solvents, and then injection to GCFID system, and also quantification, calculating of alcohol using the standard series of calibrated curve. And the method infographic for HALA certifications, that the validation method using for gas chromatography, and also the HALA verification is must be selective, precise, and accurate. We have to make sure about all our testing capabilities already validate, and to explain to our customer that our methodology is already conduct by validation, to make sure the customer can be pleased with our result. And maybe from several experience that we have from challenge and recommendation, maybe from our sites, that maybe for the follow-up in the later on, maybe for the future, we will be conduct about the LAR test method. Yeah, usually fats and oil is very important nutrients for humans, and also a lot of food industry use lard, not lard but fat to their products, and also we have to make sure that fat that they use for the fortification should be, if the halal scope, not containing lard. Yeah, so in future we will develop the lard test method. But it means the candidate test method is we have two candidate. First is the FTIR, or Fourier transform infrared spectroscopy, and maybe the LART has a distinctive AR absorption signature to do the specific molecular vibration. So we can hope that the LART can be able to detect with FTIR, because they have a specific spectroscopy. So we hope the FTIR can detect if the product have LART in it. And also for GCFID, we now work for the specific characterization regarding the saturated fatty acid that LART produce. So maybe in the future we will check about the standard for lard, and then we will try the detection of lard by GCFID using several matrices in the samples. And the challenge also of the analysis of the products regarding halal, first in for the PCR method, the extraction method is highly processed products and low level present DNA samples. Yeah, usually we also conduct in LCMS because sometimes we have samples that it's remain the low DNA and highly ultra processing food that usually the pairing of DNA is very low. And the recommendation for qPCR method to optimize and validate the DNA extraction method to its type of matrix sample, because many samples that very various matrices that challenge that we must do the validation its matrices. Second one for the LCMS about the FATWA, the impression of result, because usually we have two markers. The two markers already halal or haram. We must discuss also with the regulatory body. And also the QCM availability for LCMS method is still difficult to get in the market. And the recommendation is also important to harmonize the method use, because there are several methods in the world that conduct with several brands of LCMS. And also the challenge for the all is expansion the global halal standard, by SMIC or by maybe in, I don't know about the AOAC or about the Islamic country to do the global halal standard, and also the harmonization existing standard method for the halal. And the recommendation is strengthen the halal product assurance synergy and collaboration internationally, because we also need friends to discuss about the halal testing or halal assurance in several products. And also number two is collaborative framework involving stakeholders such as certificate bodies, industry expert, academia, and also customer for developing global halal. And we also still do with the halal consortium that held by Indonesian government by BPJPH. So for the next future, we hope the collaborative is can tight in the future. Maybe that's all about my presentation. Thank you very much for your attention. Jazakumullah khairan kathiran. I will return back to Ruba. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, our speakers, for this detailed presentation, Dr. Silvia and Dr. Dewey. And we are now having some questions in the chat box. So maybe I can start with the first one, which is from Priyanka Jain. And I believe this is for Dr. Silvia. And the question is, are there other compliance criteria in halal food other than alcohol, pork, and etc.?
Hi. Well, halal, as we saw, it goes beyond the detection of forbidden ingredients. So certifications will also focus on the entire supply chain and the management and the prevention of cross-contamination throughout it. So there are requirements that definitely go beyond testing for to analyze pork and alcohol. So we saw there's a big ritual part in the slaughter of the animal, but it covers the entire supply chain, right? From the humane treatment of the animals all the way to the end. And their requirements, it can get very technical for certification bodies, for even laboratory proficiency, for test methods. So yeah, it goes beyond. And with respect to analytes, I would like to add that, yes, we focus a lot on pork and alcohol, but there may be emerging analytes that would be of interest. For example, insects, right? We see a rise in production and consumption of insect protein. So this may be something that will be of interest for developing methods that will be relevant to halal compliance. There's other controversial products like GMOs, for example, that sometimes are interpreted as halal, haram. So Yeah, there's a lot of things beyond pork and alcohol, but pork and alcohol, yes, the main focus currently. And as Mr. Dewey explained, there are challenges related with these traditional analytes that are currently being addressed with different methods. So yeah, it's quite complex.
Thank you, Dr. Silvia. And I have another question in the chat box for Dr. Dewey. And the question is, of course, from Dr. Hamzawi. For additives, just as caramine and the pure pigment caraminic acid derived from Coachella bugs is still described as halal, even if it's derived from bugs. So, what is your opinion about this, Dr. Dewey?
Yeah. Thank you, Ruba. For carmine, carmine is usually that's pigment for food color and also cosmetic. For our Indonesian Ulama Council is already halal because it's made by bugs. Yeah, bugs is we also heard from hadith. Yeah, bugs, grasshopper is halal. Yeah, so our Indonesian government already established the fatwa MUI number 33 2011 that carmine is halal. Yeah, and also already we want to answer about the limit of detection of qPCR in our lab is 0.1%. The question from anonymous attendee that mentioned the asking about the limit of the detection of qPCR that already conduct on our lab. Yeah,
So
thank you, Ruba.
Thank you. It's 0.5.
The 0.1%. Yeah.
There's also another question from an anonymous participant. What is, of course, it is the same. What is the detection limit of DNA detection using PCR? And are there any kits based method or kits available for pork testing? Dr. Dewey?
Yeah. The kits for the pork testing, usually we have testing by our vendors. Yeah, we use waters from waters testing kit from quantitative for LCMS-MS. We use waters. And for maybe from the DNA, we use commercial kit for with optimization by our lab. Yeah, usually it conducted in several brands. Yeah, what do you call it? From the Chayagen on several etc. Usually we optimizing with our validation in our lab. Yeah, and also sometimes several vendors usually they have our own method for the markers of the LCMS-MS, and also they also have several uh, condition. Yeah, but for the practically usually we do the optimization by ourselves because regarding we are third party laboratory that we have some fortune from our testing, so we make sure that all about the kit that we use is suitable with um, our customers. Yeah, because we not can sell the analysis is too expensive. Yeah, so usually sometimes we make our method by our own optimization. Yeah, thank you very much.
Thank you. And there's
Dr. Ruba, if I may add something there. For rapid methods like industrial applications, quick verifications, like not necessarily certified lab. There are also rapid methods based on ELISA. There's even lateral flow devices for yes or no answer of presence of pork. So there's also the commercial rapid methods available that are either based on ELISA or even PCR. You can get results quickly for pork. Yeah, just wanted to add that insight.
Thank you very much. And we have also another question in the chat box, which is how about halal label to a product that has one of its ingredients as a product of microbial metabolic processes, bacteria or molds, such as lactase enzyme or cellulase? Who would like to answer it? Do you need me to repeat that again?
No, I can. This highlights one of the issues with the differences in standards, right? They will depend on what is considered halal or not, but from an analytical methods perspective, we can identify if a sample comes from a microbial or chemical conversion process. There's magnetic resonance, I think, infrared spectroscopy, mass spectrometry. So isotopic methods. So there are methods out there. There's probably costly, but it is possible to reach an answer. But the categorization of the food as halal or not, that's another question.
Thanks for the expert.
Yeah, I want to also answer about that things microbial metabolic process. Usually we have some products that use maybe maybe similar with that, yeah, like yogurt, kefir, sometimes like some things food like that, yeah. Maybe if we just check it with in terms in alcohol contents, is maybe it will clear the ethanol, the alcohol. But once again, usually we use the fatwa in Indonesian Ulama Council that maybe in kefir or yogurt is halal. Yeah, but I don't know about these things about the metabolic process bacteria of mold is very specific things. But in Indonesia, usually for yogurt, for maybe some probiotics matters is labeled halal. Yeah.
Oh, you do put the label on it. So, okay.
Yeah.
So there's a question also for our esteemed speakers about chili sauce, and that in the US it contains methanol. How happened the fermentation? So it's not really clear what the question is, but does she mean here how does it happen through the fermentation of the chili sauce, or so it seems maybe it's natural or unnaturally produced? Dr. Dewey?
Maybe yeah, maybe from our opinion about the maybe fear of methanol, because I don't know about the process, yeah. Uh, Maybe uh, how the percentage of the content of methanol in that sauce, it's small amount, 0.1% or 1%. We must know about the question, yeah, because several things that from the sauce it can appear also from the production process. Usually they use for maybe some solvents to make the CIP or cleaning in place in their production, or they use methanol, or they use ethanol, or it's mean that appear in the fermentation. We must check about uh, the the audits or the processing is we must to be checked. but the detections maybe can able do it with GCFID from headspace, because ethanol, methanol, and also n-propanol we can do it simultaneously. But why it's appear methanol is maybe very hard to answer uh, very with maybe specific explanation, because the fermentation is very complex. It depends about the uh, what is the raw material for do it for the sauces, what is the yeast, or what is some things that can the methanol is can appear in the sauce. Yeah, thank you, Ruben.
Thank you. And we have also here a question written in Arabic, but the question is that if the certification of halal, if we put it on any food, it has to be tested, laboratory tested. So this is our question. Is this a mandatory, or it can be, we can put the certification or give the certification without testing?
Yeah, that's a uh, interesting question. That's a happen in Indonesia, and not also all the halal label is conducted by laboratory analysis, because in Indonesian perspective and point of view, and also for our government objective for doing the halal certification mandatory in 2026, all about the small industry. Usually they only use the audit and then also the certification because about the price. Yeah, because about the price. But not all the halal label is conducted by testing laboratory. No. Yeah, from Indonesia opinion. Yeah, but it should. Yeah, but right now is we still obey the government, so we still follow the rules. So they it not necessary to do all about the uh, label it halal is most conducted by laboratory analysis. Yeah.
Thank you. And what about those coming by biotechnology, the GMOs, innovative, novel food? Is there any procedure to check or validate it's halal? Dr. Silvia, would you like to answer?
Yeah, well, again, I'll give my same answer. That is, we can analytically tell if a certain GMO organism is there, but then if it is considered halal or no, it will depend on the jurisdiction and the standard.
Okay. Is there now any new way to detect halal meat other than DNA analysis? Dr. Tharwat Al-Batayne from Dubai. Dr.
So I will let Mr. Dwi expand on that, but just want to point out that apart from DNA analysis, it's not a new method, it's actually an old method, but there's also ELISA, which is based on protein detection. But new methods, I would let Mr. Dwi tell us about it.
Yeah. Thank you, Sylvia. Yeah, sure. There's also by rapid test and also for with written PCR usually for halal meat. Uh, for halal meat is tricky also. Yeah, halal meat is not also contain pork and bovine, but you have to make sure about the slaughtering about in the butcher is needed to have audits in there. Yeah. But for laboratory testing, yes, we have qPCR, we have PCR, we have rapid test, because uh, for meat is the DNA is still many appear, so it will not difficult to do it even by test kit. Yeah.
Thank you. And again, Dr. Dewey or Mr. Dewey, what is your opinion if auditing was done to ensure that there is no pork or non-halal ingredients in a product? Even then, is it required to do halal testing?
Yeah. Sure. Usually, experience in several customers that we have. Usually, they do the self uh, voluntary analysis to do it. Yeah, they make sure about the raw material that they use is really use halal raw materials. Yeah, and also usually for make sure and also to trust from the customers. Usually is needed also to do the halal testing for your product. Yeah, sure.
And also another, it seems it's a very interesting subject for especially our region. And there's another one that says we need new searches and documents if insects used as a source of protein from safety aspects as allergies and from halal aspects. I think Dr. Silvia maybe can answer this.
Yeah, well, it's a comment. Yeah, I agree that we need more work related to protein sources originating from insects. It's relevant in different aspects of food safety, halal because of the different permitted insects, allergies because novel food proteins may elicit immunoglobulin, well, allergic reactions in certain consumers. There's the issue of fraud also because you may market a product saying it contains insect protein. Some consumers are attracted to that, and finally there's no insect protein. So yeah, knowing that this is coming as a huge wave of new protein sources, yeah, a lot of work is needed on this area beyond halal. Yeah.
Okay, thank you. And there's also a question in Arabic concerning the food additives that we don't know if it can be from animal origin or not, like the calcium. Are we able to detect it or differentiate the origin of the food additive?
Kalsium kita bisa nggak ya? Chondroitin? For our experience, usually I don't know what is the calcium from it. Yeah, if from bones, usually in pharmaceutical product that we already receive, they use chondroitin. The chondroitin, we able to do it with qPCR. We already validated. Yeah, and also if the content is calcium, but specific calcium from bones. We don't any idea, but from chondroitin we already validate. Yeah, thank you.
You're welcome. And there's a question also if there is a proficiency testing programs like PTs, if it's available for halal testing.
I believe,
Yeah.
I don't 100%
yeah.
sure, but I believe SMIC has something related to proficiency testing. Yes, Mr. Dweck?
Yeah, SMIC have. Yeah, SMIC have. SMIC have. Yeah, but the matrices is still just very few. Yeah, maybe we must to conduct a lot of matrices in the future. Yeah, thank you.
So there is. And how do we know the claims about the products that they are halal or free of pigs and their derivatives without conducting laboratory tests? Dr.
I will add my food safety professional experience here beyond halal. Food safety for any contaminant is not demonstrated with testing of finished product exclusively. This is something that we would do for non-compliant or suspected samples, but usually audits and certification should give you the assurance that the product is safe. So same for halal. I don't think we will need to test every single product out there to see if there's pork in any form or alcohol, but that's where certifications are there to give you the assurance through a comprehensive approach that starts way beyond the production operation. It starts from raw materials, even treatment of animals, etc. So analytical methods, of course, are very important, and it complements and is part of certification process, but I don't think every single item needs to be tested.
Thank you, Dr. Silvia. I think Dr. Eric, do you have?
Yes.
Yes, please take the floor.
Yes, thank you very much, and thank you very much to Dr. Silvia and Mr. Dewey for their presentations. I have a question related to. I heard a lot about need for methods, standardization, harmonization. So at AOAC International, we are the platform, I would say, to work on that. So, Dr. Dewey, you did not mention in your presentation about existing official methods, like, for example, AOAC. I know ISO is also working on PCR-related methods, etc. So, if you would have the opportunity to propose, let's say, one priority to work on an official method, standardized method in this topic for halal certification, to ensure that all laboratories, let's say, come to the same conclusion in regulatory decision making, as we heard earlier. Also, like, for example, certainly the Arab section, but certainly also the Southeast Asia section, has a working group on method harmonization. So if you would like to have the opportunity to propose, let's say, a priority to work on a future officialization of a method, what topic would you then propose currently and as the highest need for standardization? Thank you.
Thank you, Dr. Eric. This is a great overview. And there's also another question, what is the criteria for distinguishing between, is there a percentage of alcohol can be naturally occurring in food like juices and still halal?
I answer maybe from Indonesian Ulama Council, the threshold is 0.5%, 0.5%. But from the juices, yeah, that's maybe we can make sure also from the audits, yeah, because if the fermentation is not do it for Homer that I mentioned in my presentation, I think it's still halal, yeah, because but it depending on the purpose, If the juices is like a grapes, it can be wine. Wine is Homer is haram. But from juices, yeah, it will be contain alcohol, but from our Indonesian side, the threshold is 0.5%. Yeah, thank you.
Sorry, if I may, still my question was not answered by Mr. Dewey or Dr. Silvia. What would be the first priority for a need for standardization from their side if they would have the opportunity? If this is possible to.
I would start beyond the test methods. I would like to see a unified halal standard, and that would guide where do we need, if we start with a unified standard that has clear limits, for example, of alcohol content, naturally produced, industrially added, gelatin, etc., then from there I would go into the methods that would be selected or most adapted to this, to prove the standards, and that could be used in certifications. But about the technical challenges, I would leave that to Mr. Dewey.
Thank you for the share. That's why we have three kinds of analysis. Yeah, that's why, and also our homework about the determining the LART, and also a lot of homework that we do the collaborative studies. Usually, right now in Indonesia, in Halal Consortium, we have collaborative study by QCM from our halal product assurance organizing body. Yeah, they have the QCM, but still in the very small matrices, kind of a matrices. Usually just for meat, processed meat. That's mean the QCM, and they test to several laboratories that already declare they can be analyzed halal to make sure the performance of the laboratory. Yeah, because uh, in this also we have to homework the priorities about the markers of the LCMS MS is also important. Yeah, because the markers is conducted by high resolution matrices, high resolution mass from TOF. That it means the markers is also very important. Yeah, the markers is must be standardized is all over the world. Yeah, what is the maybe I don't know about the swines, but what species that they use about that? Yeah, the markers about the matrices, about the maybe from the PCR side. How about the temperature? How about the nailing? About the CTs? That is maybe a lot of things that must be harmonized regarding to the analysis, Eric. Yeah, I hope that's can answer, and also uh from the OASIS side, as I think that's a big challenge also. If we can do the harmonization about the method of the regarding the halal detections, just not the three things. Yeah, maybe one things for PCR is quite challenging, quite challenging. The matrices, the not all the lab have the qPCR. Usually they only have PCR, or even ELISA. That you have to make sure about the capabilities on the whole world laboratory regarding the halal assurance in the world. Yeah. Thank you, Eric.
Thank you.
Um, Dr. Eric, I think also Dr. Amin Goussou has a question. Dr. Amin, please take the floor.
Thank you, Dr. Ruba. It's actually it's not a question. I would like to second Dr. Eric's comment and emphasize on the need for methods harmonization. And here I would like to grab this opportunity also with to have a proposal as like a conclusion from this webinar and from this collaborative work between our two sections. So I have a proposal to be creating like a common working group between our two sections and be working on defining priorities for method harmonization. We have Dr. Ximping also with us, who can second me also about regarding this proposal. So thank you, Dr. Eric, for sharing your insights with us. And why not having our working group between our both sections to be working on defining priority for methods harmonization and then be working on harmonizing methods between our two regions. Thank you so much.
Thank you, Dr. Amy. I couldn't agree with you more on this. Yeah, I second you as well, together with Eric. That hopefully we can work together to create an initiative. Maybe start with a working group or the better panel to discuss how we are going to proceed from here after this webinar.
Thank you. I think this is a great outcome from the webinar today, to start on something that can harmonize all the procedures we have in our region. So we have to be quick because we are running out of time. I do have three questions. One, is there a test method that can identify if the alcohol described in food is from a natural fermentation or synthetic alcohol added during manufacturing? I think we did answer this question, or would anybody like
Yeah.
to?
Maybe right now is we cannot from our opinion, from our experience, we cannot do the several difference, yeah, between the maybe alcohol from fermentation, alcohol is from the unorganic ways, that it's still difficult. Maybe I don't know. Next, if needed with NMR, needed with several sophisticated techniques, but once again, that will make the testing is quite expensive. Yeah. But now it's containing for the right now that we know that we just do the FID detection to the alcohol content. Yeah. But make a difference is we have not idea from our side. Yeah. Thank you.
You're welcome. And there's another question. What is the Islamic ruling on monk fruit, as it contains a percentage of natural alcohol and is used as a sweetener because it contains a low calorie amount of sugar? Monk fruits. So, Mr. Dewey.
Monk fruit. Okay. That's very new. Do you know, Dr. Sylvia? No?
I was going to say me neither. I'm going to Google it. No, I don't know.
I don't have any idea for monk fruit. I'm so sorry.
We'll send the answer. We'll definitely will check and send the answer to our participant.
Yeah.
Last two questions. There is many. Okay. What is the detection limit for porcine gelatin in capsules? If it is below the detection limit, then what compliance should we give? Dubai Central Laboratory. So this concerns the capsules in medical capsules and the food additives.
Can you repeat again, Ruba? I'm so sorry.
What is the detection limit for porcine gelatin in capsules? If it is below the detection limit, then what compliance should we give? And this is the Dubai Central Laboratory.
Yeah. Okay. Usually, we still yeah, our detection limit is 0.1% for the I mean the capsule, yeah, or the soft capsule by the LCMS-MS. Once again, we state in our certificate, because we are third-party laboratory that not give the halal. Usually in our certificate we just state negative porcine, and we state also the our LOD and LOQ 0.1%. So usually that's information that will be seen in our halal regulation body in Indonesia. Oh, the laboratory analysis is all below 0.1%. For the detection is depends about the countries and also the rules. Yeah, that Dr. Sylvia already mentioned that usually in Indonesia 0.5, in Malaysia is 1%, and it's depending on your countries. But to me, if the result is below the LOD and our LOD is 0.1%, we state in our certificate is negative. Not halal, yeah, it's negative. Yeah. Thank you.
Thank you. And this is also a last question for Dr. Dewey. For Mr. Dewey, what is the future steps that can help the Islamic countries to unify the halal concept, regulations, and analytical methods? And this is from Dr. Thuraya. I think maybe the working group that was mentioned today would help
Sure.
in unifying all these. So we'd like an answer also.
Maybe Dr. Sylvia, you have any?
Yeah, it's a big question for Mr. Dewey. I think, like Dr. Ruba said, this is should be addressed as a community, right? A regulatory, analytical, religious. There's so many aspects related to halal food. So I don't want to leave Mr. Dewey to answer this question by himself.
Yeah.
But I think the best idea is to implicate all the stakeholders and gather their opinion, right, on this topic. It's quite complex. That's why we don't have a unified halal standard, for example. It's not because people haven't thought about it. It's just there's so many interpretations, variations. So So yeah, I would say one of the main steps, I keep saying this, would be to have a unified halal standard. This would guide then the methodological aspect. But that's my point of view. I'm sure others have other perspectives, and that therefore the importance of working as a community, right? And this the opportunity to do this specifically now in the Arab and Southeast Asian regions that are really concerned with this subject.
Yes, for sure. I will have to maybe last the comments about the harmonization and also all the stakeholder, yeah, maybe in Indonesia, in Ministry of Religion, our health accreditation body, our uh, technical committee regarding the analysis, and also about the auditors. The competency of auditors is really, really important because they look the process about the halal certification until the raw material and also the process in the factory. It's very important to we know, and also they already well trained regarding the audits of the halal certification or halal process. And once again, the harmonization of what this uh, uh, kind of assurance of the halal uh, Indonesia, it's still homework. Yeah, still homework about the implementation, and also or even in the Arab section or even in the world is still we need to have to many um, technical and also religion aspects to conduct the harmonization and also the group discussion regarding of this. Yeah, maybe I think this webinar is one of good condition that we can help also in the future regarding in our section in Southeast Asia, maybe in Indonesia, and also for your section in Arab and European and so on. Thank you. Thank you very much.
Thank you very much. Dr. Amin, would you like to add something? Because you brought up this great idea about...
Thank you, Dr. Ruba. No, I just want to thank everyone. If you allow me to thank Mr. Dwi, to thank Dr. Silvia. Thank you first, Dr. Ruba, for the amazing moderation of the session today. I would like to thank Dr. Eric for all the support since the early beginning of this idea. And thank you, of course, to Dr. Ximping for all the support from our sister section. So thank you all.
And thank you, Dr. Eric, for this great initiative, really. And I would really like to extend my sincere thank you for all our speakers and for your really valuable insights. And this has been really an incredible, informative session and very interesting to our region and our community. I am sure many of you have gained a deeper understanding of the methods of analysis employed in this field of halal food certification. Thank you again, our speakers, for your experts' insight, and to everyone for your participation. And kindly note that the presentation would be available soon on G4's website, and the full recording in both languages, Arabic and English, on G4's YouTube channel. We all look forward to seeing you all again at our next webinar. Assalamu alaikum.
Thank you.
Thank you.
Thank you. Bye-bye.
Bye-bye.
Thank you, Eric. I'm in. Dr. Silvia. Thank you very much. Dr. Ruba, thank you.
We share with pride for a world where health is on our side. Collaboration and science as pillars. G-Force, we aim for rigor. G-Force together.